Hi, and welcome to my presentation on the metabolic diversity of microbes. So we will start with phototrophy and photosynthesis. Remember that phototrophy is getting energy from light. So phototroph is an organism that uses photosynthesis to make ATP from light. So photosynthesis is a way to make energy from light, but it's also a type of carbon fixation. Carbon fixation means that you're taking inorganic carbon and putting it into an organic format. So you're changing an inorganic molecule that contains carbon into an organic molecule that contains carbon. Most organisms cannot use inorganic molecules as a source of carbon, but they can use organic molecules as a carbon source. And every organism needs a lot of carbon to build all of their organic molecules. So in carbon fixation, you are fixing inorganic carbon into an organic format that anybody can use, which is why they call it carbon fixation. In photosynthesis, the inorganic carbon that you're starting with is carbon dioxide, or CO2, from the atmosphere. In the process of photosynthesis, carbon dioxide is reduced to make organic compounds like glucose or other sugars, and then anybody or any organism would be able to use those compounds as a source of carbon for building their own biological molecules. And it takes a lot of energy to do that to carbon dioxide. You have to put in a lot of energy to get carbon dioxide to go to glucose, for instance. That energy is coming from light. So most organisms that do photosynthesis are photoautotrophs. They're phototrophs because they use light for energy, but they're autotrophs as well because they're using light to fix carbon and get carbon from an inorganic source. You also do have some photosynthetic bacteria that are not autotrophs, that are heterotrophs instead. So they are not using carbon dioxide in photosynthesis. They'll be using some type of organic carbon containing molecule instead. But the majority of phototrophs are photoautotrophs who are using photosynthesis to reduce carbon dioxide to an organic compound like glucose. Bacteria are the first organisms to have evolved photosynthesis. There's a lot of different groups of bacteria that contain phototrophs. That would include the heliobacteria, acidobacteria, cyanobacteria. Those are all phyla of bacteria, which are large groupings of different bacterial species. You also have the green sulfur bacteria, the green non-sulfur bacteria, the purple sulfur bacteria, and the purple non-sulfur bacteria. So all of those different groups are able to do photosynthesis, and they each have little differences in the specific ways that they're doing it. Actually, most of those organisms are doing anoxygenic photosynthesis, which is the photosynthesis that does not create oxygen. Only cyanobacteria are able to do oxygenic photosynthesis and create oxygen through photosynthesis out of the bacteria. Then, of course, you have eukaryotes that do photosynthesis as well. That would be plants and algae. They actually gained the ability to do photosynthesis from bacteria by consuming a cyanobacterium. Far back in the depths of time, you had some early eukaryote that consumed a cyanobacterium, and then that cyanobacterium eventually turned into a chloroplast, which is the organelle that eukaryotes use to do photosynthesis. So since eukaryotes got their photosynthesis from cyanobacteria, they do oxygenic photosynthesis as well, photosynthesis that produces oxygen. Photosynthesis is an extremely important process to life for two reasons. First, in most food chains, photosynthesis is kind of providing the energy, basically, that fuels the entire food chain. And also, for most of those food chains, it is the greatest source of fixed carbon, or maybe the only significant source of fixed carbon. So this diagram here is kind of illustrating that, where you have energy from the sun coming in, and these plants are doing photosynthesis using that energy, producing lots of glucose and other organic molecules using that. Then you have animals that eat those plants, They take in the organic molecules, they use all that energy in those organic molecules to make ATP, but that energy is ultimately coming from plants, which means it ultimately came from the sun. Then you have maybe some predators that eat those herbivores, and maybe even another level of predators that eat the first level of predators. Maybe different levels in different food chains, but in each case, energy is transferring from plants up to the next level of the food chain and then to the levels above. So the plants are taking light energy and using it to make glucose. This rabbit is taking in that glucose when it eats a plant and using that to make ATP, also using that to build its own organic macromolecules. And then when the snake eats the rabbit, it's getting all of those organic molecules from the rabbit and using them for energy. All of that energy that is ultimately coming from the plants, which got the energy from the sun. The same thing for fixed carbon. All of the molecules, all of the organic molecules in all of these animals those are all made using carbon that was initially fixed by these plants from carbon dioxide in the atmosphere. So if you were to destroy those plants, you would destroy the entire food chain. There would be no way for non-phototrophic organisms to get energy ultimately from inorganic sources or to get carbon. We can divide photosynthesis into two parts. First, you have the light-dependent reactions, and then you have the light-independent reactions. So the light-dependent reactions are called that because they require light in order to occur. They can also be just called the light reactions for short. The main process that's going on there is photophosphorylation. So photophosphorylation is really similar to oxidative phosphorylation that you might remember from when we went over respiration in chapter 3. The main difference is that you're using light as your source of energy for an electron transport chain rather than electron carriers. Most phototrophic bacteria are going to have little folds of their membrane, so their cell membrane is going to kind of fold inward and invaginate into the cell, and you'll have little folds that it forms there. And it's in those folds that you have the photophosphorylation process occurring. So it's actually an electron transport chain that is located in those membrane folds. In a eukaryote, eukaryotes would use chloroplasts to do photosynthesis, and each chloroplast has membranes inside of it, membranes that are formed into little discs. Each disc is called a thylakoid. So it's in the membrane of those thylakoids that photophosphorylation is occurring in a eukaryote. And then this diagram is showing some of the different types of membrane folds that can be formed by the cell membrane in phototrophic bacteria. So for this guy, all these little circles are formed by membrane that's actually folded membrane down from the cell membrane. So this is all actually one single membrane, not separate membranes inside of it. Photophosphorylation is always going to require some type of an electron donor, and it requires light. For organisms that are doing oxygenic photosynthesis, that would be the cyanobacteria, and for eukaryotes, the electron donor that they use is water. And when they remove the electron from water, they're going to create oxygen. For anoxygenic phototrophs, it's going to be a different electron donor. And photophosphorylation is going to create ATP and some type of electron carrier. Sometimes that's NADH, sometimes it's NADPH, sometimes it's something else like ferredoxin, which is a protein that can be used to carry electrons. So it says NAD and then in parentheses pH because a lot of the time, or usually that is, it's going to be either NADH or NADPH depending on which organism we're talking about. Then of course, if you used water as your electron donor, you're going to be creating oxygen as well out of photosynthesis. So this diagram is showing a summary of photosynthesis for a leaf. So this would be a very similar process as what is occurring in cyanobacteria. At the top here you have the light reactions, and at the bottom is where you have your light-independent reactions, which we'll mention in a moment. But light is going to be coming into the chloroplast, and it's going to be used in the light reactions to fuel, to power, removing an electron from water. So water doesn't normally just give up its electrons. It's a pretty stable molecule. You need to put in a lot of energy to actually strip an electron off of water. That energy is coming from this light. Once the water has lost its electron, it's going to combine with another water that has lost its electron to form oxygen. Then that electron is going to travel through the electron transport chain that you have in these light reactions. It's going to be used to create an ion gradient. That gradient, an ion concentration gradient, I should say, that gradient is going to be used to make ATP, and at the end your electron is going to be added to an electron carrier, such as NAPD+. No, that is wrong actually. It should be NADP+. I was right. Twice. Once you've added that electron to NADP+, that gives you NADPH. And then the NADPH and the ATP are going on to the light-independent reactions, which are the reactions where you're actually fixing carbon. So now for our light-independent reactions. Those are also called the dark reactions because they can happen in the dark. The light reactions have to happen in the light, of course, since they require light. These are the reactions where you're actually fixing carbon, taking that inorganic carbon dioxide and converting it into an organic molecule like glucose. There's actually a number of different pathways that exist. So there's multiple different versions of a dark reaction that different microbes can do. The most common one is the Calvin cycle. So the Calvin cycle is what is performed by cyanobacteria, also a lot of other bacteria, and this is the one that's performed by eukaryotes, the plants and algae that do photosynthesis. You also have some other pathways that specific bacteria are going to use instead of the Calvin cycle. For all of these different pathways, you're usually using carbon dioxide as the ultimate source of inorganic carbon, and they also require a lot of ATP and electrons from electron carriers. Usually those electron carriers would be NADH or NADPH, but it could also be ferredoxin. So in the dark reactions, you're using all that ATP and NADPH or NADH to reduce carbon dioxide. So you're adding electrons to it. That requires a lot of energy. You're also ultimately going to be linking those carbons and the carbon dioxide together to build some type of organic molecule, usually a sugar like glucose. At the end, you're going to get out your organic molecule, your glucose. You're also going to get your electron carrier that is now oxidized. So it has lost its electron, so now it's NAD+ or NADP+. That NAD+ or NADP+ is going right back to the light reaction, so it can be recharged with more electrons. Then the glucose is going to go on. You could use it to build cellular structures, or you could use it in respiration to make even more ATP. Actually, most of the ATP that you make in photosynthesis needs to be used in these dark reactions to create the glucose. That's a very ATP expensive process. So a lot of the time, these phototrophs are going to turn around and use the glucose that they made in respiration to get ATP. So in our diagram here, here's the light reactions, and here's our dark reactions, mostly the Calvin cycle. So out of the light reactions, you're getting ATP, and you're getting an electron carrier, such as NADPH. Then for the dark reactions, you also need carbon dioxide, which would come in as a gas. Then you're going to take those electrons from the NADPH, or whichever electron carrier you're using, and transfer them to the carbon dioxide. You're also ultimately going to be linking these molecules of carbon dioxide together in a process that requires a good amount of ATP, and you're going to be getting some type of sugar on the other side. So all this carbon dioxide is being converted to sugar. Your NADPH, or whichever electron carrier you had used, it has lost its electrons now, so now it's NADP+, or NAD+, or ferredoxin without an electron on it. And that's going right back to the light reactions. so that it can be recharged with more electrons. And also, once you've used that ATP, it is converted into ADP and a free phosphate. That's also going back to the light reactions, so the phosphate can be reattached to ADP, giving you back ATP. So now we'll focus in on photophosphorylation and go through it in a bit more detail. Photophosphorylation requires pigments. A pigment is just a molecule that is able to absorb light, which specifically means that it's able to absorb photons. So different organisms will use pigments for different purposes, but you're always going to need it for any type of organism that is responding to light or doing something with light. It's that pigment that's actually absorbing the light and lets you do something with the light or lets you respond to it. For phototrophs that use their pigments to do photosynthesis, they are going to have these giant complexes called photocomplexes that have a lot of pigments in them and also have protein in them. So it's pigments basically structured with this protein scaffold. Those photocomplexes are going to be embedded in their cell membrane or in one of those membrane folds that they have. The benefit of the photocomplexes is that it allows you to concentrate light. So sometimes bacteria are living in a place where there is light, but there's not like a lot of light. Maybe they're kind of under a bit of water. So there's still light getting down to them, but it's not like as bright as daylight is. So they're going to need some way of concentrating the light energy so that it's actually enough to strip an electron off of the electron donor that they're using for photophosphorylation. In photocomplexes, you can have a bunch of different pigments that are all absorbing these kind of low energy photons and then funneling the energy into the center of the photocomplex for photosynthesis. So a photocomplex is what you have right here. Up here you have a couple different pigments that bacteria use. So these pigments would be embedded in the different proteins of the photocomplex. So light energy, when it strikes any part of this photocomplex, it's going to be ferried into the center, the reaction center, right in the middle. That's where you're actually going to have that light energy stripping an electron off of the electron donor. Then that electron will enter an electron transport chain. Some of the main pigments that bacteria use for photosynthesis are chlorophyll and bacterial chlorophyll. Chlorophyll is used by cyanobacteria only, actually, but it's also used by eukaryotes since eukaryotes got their photosynthesis from cyanobacteria. Chlorophyll is able to absorb red and blue light wavelengths, which means that it reflects green light, so that's why it looks green to us, and that's why plants look green. Any pigment is going to have kind of a range of wavelengths that it's able to absorb, so it needs to absorb a photon that is in that right wavelength, and if a photon comes in at the wrong wavelength, it's just going to bounce off. Bacteria that are not cyanobacteria are usually going to be using a bacterial chlorophyll pigment, and there's several different ones that they could be using. It's very similar to chlorophyll, but just a little bit different. In this diagram here, you can actually see chlorophyll and a bacterial chlorophyll. So you can see that they're really, really similar. They just have a couple differences up at the top of the molecule there that are highlighted. Which wavelengths bacterial chlorophylls will absorb and therefore which color they have is actually varying depending on the specific version of that pigment. You also have some phototrophs that use another type of pigment, the phycobiliprotein. That's actually, the pigment there is bilin. You have a bilin pigment and then it's complexed with proteins to give you a phycobiliprotein. So you have some cyanobacteria that use those pigments and also red algae that use those pigments. Those are not going to be the main pigments that they're using, but they would be used to collect extra light. So you might have in this photocomplex some phycobiliproteins around the edges of it. They're going to be catching extra light and ferrying it into the center where you would have your chlorophyll. You have a couple different colors for phycobiliproteins. Some of them are red and some of them are blue. In addition to those pigments that are being used to absorb photons for photosynthesis, any phototroph is also going to have other pigments too, which are generally going to be carotenoid pigments. But those pigments are not actually designed to absorb photons for photosynthesis. They have a protective role. So they're basically designed to protect the cell from really high levels of light. If you have really high intensity light and that strikes oxygen, it'll actually generate reactive oxygen species, or ROS, which would be dangerous for the cell. So any phototroph, basically any phototroph, is going to have some of these carotenoid pigments that are designed to absorb those really high energy photons so that they do not make reactive oxygen species. Carotenoids have a lot of different colors, and there's a lot of different carotenoid pigments. They usually are going to absorb blue the most strongly out of any type of any wavelength of light, so you don't find them in the color blue, but they have a lot of other colors. They would actually be giving color to most of the fruits and vegetables that you're familiar with. So here's a diagram of a carotenoid pigment. So you can see it actually looks quite different from the chlorophyll and bacterial chlorophyll pigments. And all of these veggies and fruits up here are getting their color from different combinations of carotenoid pigments. So you're actually very familiar with them, with seeing them anyways. They can be useful for you when you eat them because they're able to act as antioxidants and prevent damage to your cells from reactive oxygen species. Different carotenoids are actually going to be better at that or less good at that depending on, which specific carotenoid it is. So different carotenoids will have different health benefits for you. In general, it's a good idea to kind of mix the colors of the veggies that you're eating so you make sure you're getting a range of those pigments and of the other vitamins that the vegetables have. Let's see, what else was I going to say about carotenoids? Oh, right. So actually, if you take a plant and remove its carotenoid pigments, maybe by genetically modifying it so that it can't make those pigments anymore, actually what's going to happen when you stick that plant into bright light is that it's going to die. It'll start off as a nice green plant. It's green because it has a lot of chlorophyll, but as time passes, the light is going to be creating reactive oxygen species, and those ROS are actually going to damage the chlorophyll. So eventually, the plants are going to lose their chlorophyll. They'll lose more and more of it, and they'll turn brown, and when they're not able to do photosynthesis anymore for energy, they will die. So the carotenoid pigments are really important to allowing organisms to do photosynthesis, even though they don't directly participate. And then the final image here is a pool that is full of purple bacteria, giving it that kind of pinkish purplish color. And those bacteria are getting their color from the carotenoid pigments that they have. So basically any photosynthetic organism is going to have some type of carotenoid pigment in it. So first we'll talk about the details of anoxygenic cyclic photophosphorylation. Any type of photophosphorylation is going to include two parts, an electron transport chain and ATP synthase. And they work together very similar to how they work in oxidative phosphorylation and respiration. This is anoxygenic photophosphorylation because the electron donor is not water, so it does not create oxygen. And it's cyclic because the electrons are actually going to be traveling in a little circle from where they started back to where they begin. And the diagram there is it's a diagram of the photophosphorylation chain in purple bacteria. So first with the electron transport chain, as you can see, it's a pretty simple and short electron transport chain, and it is quite simple and short for most of the bacteria that do photosynthesis. You're starting with your photocomplex right here. So photons are going to be hitting this photocomplex. They're going to be absorbed by the pigments there, and the energy is going to be funneled into the center. In the center of the photocomplex, you have a couple of special bacterial chlorophyll pigments, the P870 pigments. So when the photon energy hits those pigments, it's actually going to be strong enough to strip an electron off of them. That electron is then going to enter the electron transport chain, First it travels on a quinone, then it goes through the membrane to this other protein complex here. Ultimately, it's going to be transferred to a cytochrome molecule and travel back to the same photocomplex that it came from. This electron is going to be added back to the P870 bacteria chlorophylls. So it's starting there with the bacterial chlorophylls, goes around the electron transport chain in a little circle, and ends up back at the special bacterial chlorophylls. As those electrons are moving on the electron transport chain, hydrogen ions or protons are going to be pumped across the membrane from the inside of the bacteria to the outside. And you can see that here that's actually happening because of the quinone electron carrier. So when that quinone picks up electrons, it doesn't want to just have electrons, it wants to have hydrogens on it. So it needs to grab a couple of protons from the cytoplasm to make sure that it balances out 2 electrons with two protons to make 2 hydrogens. But then when it's going to drop those electrons off at this other protein complex, that protein complex can only take the electrons, it cannot take full hydrogens or the protons. So that means the protons are going to get kicked off and they end up leaving to the outside of the cell. So ultimately, light comes in. The electron donor here is your bacterial chlorophyll. So the energy of this light is used to strip an electron off of the bacterial chlorophyll. That electron travels through the electron transport chain, moving in a circle back to the same bacterial chlorophyll. As it's traveling, protons are being pumped across the membrane to the outside of the cell, and that creates a proton motive force. And then you have ATP synthase allow those protons to flow back across the membrane down their concentration gradient, and uses the energy of their movement to add a free phosphate to ADP, which gives you ATP. Then this diagram here is showing you kind of the reduction potential of the different electron carriers that are involved in this cyclic electron transport chain. So first you start with your central bacterial chlorophyll, which is very bad at donating electrons, but very good at accepting them. But once it's been energized by a photon, it becomes the opposite. It becomes very good at donating electrons and bad at accepting them. So after light energy energizes that central bacterial chlorophyll, it's going to donate its electrons, and they're going to be transferred through the different carriers and proteins of the electron transport chain. At each step, the ability of the next carrier to accept electrons gets a little bit better, and the ability of that carrier to donate them gets a little bit worse. So you're always passing those electrons off from someone who's a little bit worse at donating them to someone who's a little bit better at accepting them, and so on until they get back to the original bacterial chlorophyll, which now is not energized by light anymore, which means that it's very good at accepting electrons and bad at donating them. So it's going to accept those electrons, and then the cycle will repeat. So there's different versions of that basic scheme for the different types of anoxygenic photosynthesis, or I should say for anoxygenic photosynthesis in the different bacteria that are able to do it. So each one is going to use a bit different electron transport chain, different proteins, different electron carriers, different pigments, but the basic process is going to be the same for all of them, where you have the electron donor is a pigment. So light comes in, it strikes that pigment then is energized and donates its electrons. The electrons travel on the electron transport chain. The energy of their movement is used to pump hydrogen ions, creating a proton motive force that is then used by ATP synthase to make ATP. So that's one piece of what we're getting out of anoxygenic photosynthesis in terms of the light reactions. But we also need to get an electron carrier out. So that could be NADH, could be NADP+, or it could be something else like the protein ferredoxin. For the purple bacteria that we just looked at, the way that they add electrons to electron carriers is basically that as these electrons are traveling in a circle through their electron transport chain, some of them will be diverted and added to NAD+, which gives you NADH. That process actually requires energy because NADH or I should say NAD+, is not as good at accepting electrons as the carrier that the electrons would be on at that point, which is quinone. So quinone is better at accepting electrons than NADH, or I should say NAD+. Quinone is better at accepting electrons than NAD+, which means it's not natural for quinone to give electrons to NAD+. It would like to accept electrons from NAD+, not give them to it. In order to get the quinone to give electrons to NAD+, you're going to have to put in some energy. So this process is called reverse electron flow because it's requiring energy to make the electrons go from a molecule that is better at accepting them to a molecule that is less good at accepting them. So some of the ATP that's generated by the photophosphorylation process in purple bacteria might be used to add those electrons to NAD+, giving you the NADH. For your other anoxygenic phototrophs, they use a similar process, but it's actually not as well understood. So here you have examples of the electron transport chain for green sulfur bacteria and for heliobacteria. And you can see that overall it's quite similar. You have a pigment that is being energized by light. It donates its electrons to the electron transport chain, and then those electrons start flowing on the chain and ultimately end up back at that same pigment that they came from. However, actually the situation is a little bit less clear because you have this dotted line here with the question mark on both of these guys. So that means that we actually don't know what happens between here. When the electron, like we know the electrons go to this carrier and we know that they end up on this carrier, but we don't know how they get there. Which means even though this looks like a cyclic chain, it actually might not be cyclic because we don't know what's happening in this part. Also, these guys are not using NADH, they're using ferredoxin. So they're going to end up donating some of their electrons traveling on the electron transport chain to the ferredoxin protein, which will then serve as an electron carrier. No matter how you're getting those electrons onto an electron carrier, the purpose of that is to send that electron carrier with its electrons out to wherever you're doing the dark reactions, which for these bacteria would just be in the cytoplasm of the cell. And then those electrons are going to be used to fix carbon in the dark reactions. So that was anoxygenic photophosphorylation. Now we'll look at oxygenic photophosphorylation, which is usually non-cyclic, meaning that the electrons do not travel in a circle. They have a starting location and a different ending location. This would be the type of photophosphorylation that you find in cyanobacteria and in eukaryotes. So we're not calling the photocomplexes anymore for oxygenic photophosphorylation. Instead, we call them photosystems, but they're basically the same thing. It's a complex of lots of pigments and proteins. And the job of all those pigments and the proteins is to capture light and funnel it into the center into a special pigment that is going to donate electrons. Another difference is that the pigment that we're using here is not bacterial chlorophyll. Since we're in cyanobacteria, the pigment we use is chlorophyll. So this is your kind of larger electron transport chain for oxygenic photosynthesis in cyanobacteria or eukaryotes. This is one of your photosystems, and this is the second one. Actually, this photosystem is called Photosystem 2, and this photosystem is called Photosystem 1. That's because the second one was actually discovered first, so they just called it Photosystem 1. And then after that, they discovered Photosystem 2 and realized that it actually comes first in this process. So it's a little bit backwards. 2 comes before 1. So in this photosystem 2, you're going to have light energy coming in. It's going to be striking the pigments in this photosystem. The energy from those photons is going to be transferred into the center of the photosystem, where it's going to ultimately be funneled to a special chlorophyll pigment, the P680 pigment. Once this pigment is energized with the light energy, it's going to have an electron removed from it. So it's going to donate an electron to these proteins that are part of this photosystem that can carry electrons. Once that happens, this pigment is actually going to take an electron from water. So it's going to use the last of the energy that it has from that photon to strip electrons off of water and replace the electrons that it lost. When that happens, you get O2 as a result, or oxygen as a result. So actually, technically, you need to strip 2 waters. You need to strip the electrons off of 2 waters to get 1 molecule of oxygen. Then these electrons that are on the electron transport chain that came from this pigment, they're going to be transferred from the different electron carriers on the chain through all these different protein complexes until they eventually get to photosystem 1. Once your electrons reach photosystem 1, you're going to have more photons coming in. Since this is a photosystem, it also has pigments. So you're going to have these light energy photons being absorbed by the pigments in this photosystem. That energy is going to be transferred to a central pigment, the P700 chlorophyll. And at the same time, electrons will be arriving at that chlorophyll from the electron transport chain. So that energy from the photons is going to be transferred to these electrons that are traveling on the electron transport chain and passing through this chlorophyll. So now the electrons are getting a big energy boost. So they go up to being full energy, high energy electrons. And at that point, after they get through photosystem 1, they're going to exit the electron transport chain and be added ultimately to NADP+, giving you NADPH. So that's the path of the electrons, starting out from this pigment in photosystem 2, traveling on the electron transport chain, ultimately to photosystem 1, where they pick up another energy boost and end up on NADP+, making NADPH. Then once this first pigment in photosystem 2 has lost its electrons, it's going to take new ones from water, which gives you oxygen. The other thing that's happening here, of course, since it's an electron transport chain, is that you're pumping hydrogen ions or protons. So you can see there's different points here on the electron transport chain where you're picking up hydrogen ions from one side of the membrane and transferring them to the other side. So this diagram here is actually showing all of this occurring in the membrane of a thylakoid inside of a chloroplast. If this was a cyanobacterial cell, then of course these protons would be leaving to the outside of the cell, and this would be the inside of the cell. Since what we're looking at is a chloroplast thylakoid membrane here, this here would be the inside of that thylakoid membrane, and this would be the outside space, which is the stroma of the chloroplast. But wherever you are, whichever membrane this is, you're pumping hydrogen ions across that membrane, and that creates the proton motive force. And then again, your ATP synthase is going to use the proton motive force to create ATP by allowing hydrogen ions to flow down their concentration gradient back across the membrane, and then using the energy of their transfer to add a free phosphate group to ADP, giving you ATP. So that's your basic scheme for oxygenic photophosphorylation. It's generally a non-cyclic process like that, where electrons are coming from a pigment, those electrons are replaced on that pigment by water, and then those electrons are being dropped off on an electron carrier, NADP+, not being dropped off back on the same pigment that they came from. But it's actually possible for cyanobacteria and eukaryotes to switch to doing a cyclic form of oxygenic photophosphorylation if they need to. The reason why they would do that would be if they need ATP more than they need electron carriers for carbon fixation. So if they're doing the non-cyclic version of photophosphorylation, then they're making ATP, and they're also making electron carriers to be used later in the Calvin cycle to fix carbon. That process of the Calvin cycle, that's going to use up the electron carriers and most of the ATP that they're making from photosynthesis. But sometimes they might find themselves in a position where they do need ATP, but they don't really need to fix more carbon right now. They would have maybe enough carbon fixed already. And if that's the case, then they might not want to make those electron carriers for the Calvin cycle. They might want to save that energy and make extra ATP. and use that ATP for whatever other purposes they need. So they're going to, if that's the case, they're going to switch to cyclic oxygenic photophosphorylation, which is shown right here. So your electrons here are actually traveling from the pigment, from the special chlorophyll pigment on photosystem 1. They're being stripped off that pigment by light energy, and then they're traveling in a circle on the electron transport chain and ending up back on that same pigment. So this is more similar to the situation in your anoxygenic bacteria that are doing cyclic anoxygenic photosynthesis, where the electrons come from a pigment, they circle around on the electron transport chain, they come back to that same pigment. Also, usually these guys are going to be doing oxygenic photophosphorylation, meaning that when it's non-cyclic, they are taking electrons off of water to replace the electrons that are removed from their pigments, and that creates oxygen. However, it's possible for some cyanobacteria and some algae to actually switch to an anoxygenic version of photosynthesis. So they would be using, instead of photosystem 2 to start, they would use just photosystem 1. And instead of replacing electrons on that pigment with electrons that came from water, they would take the electrons from hydrogen sulfide, which is H2S, or from molecular hydrogen. And then let's see, the final diagram over here is showing you the reduction potential of the different pigments and electron carriers and proteins of the electron transport chain for non-cyclic oxygenic photophosphorylation. So we would start here at photosystem 2, right here. Your light energy is coming in. That energizes this pigment. It takes it up to a really high energy so that now it is able to donate electrons. So it starts off not being able to donate electrons, but being good at accepting them, and it ends up being very good at donating electrons. So those electrons are then going to leave that pigment and hop on to the different electron carriers of the electron transport chain, moving through the chain until they get to photosystem 1 right here. At that point, you have more light energy coming in and striking the central pigment, which takes it from being a bad electron donor again to being a good electron donor, and it energizes those electrons that it's carrying. Then those electrons will continue on the electron transport chain until ultimately they make it to NADP+, forming NADPH. Okay, so that is the phototrophy part of photosynthesis. Now we have the autotrophy part, which would be the dark reactions, where you are actually fixing carbon dioxide, usually using the Calvin cycle. So first, a basic overview. Most phototrophs are autotrophs, but they are not all autotrophs. Some of your phototrophs are able to get their carbon from an organic molecule and would not be using the Calvin cycle or any other pathway to fix carbon. But most phototrophs are autotrophs. They're going to be using their dark reactions to fix carbon, taking in carbon dioxide, putting out some type of organic molecule like a sugar, like glucose. Most of these guys are going to be using the Calvin cycle. So all of your oxygenic phototrophs use the Calvin cycle. So that's cyanobacteria and all of your eukaryotes who do photosynthesis. Also, some of your anoxygenic phototrophs will use the Calvin cycle as well. So specifically the purple bacteria will also be using the Calvin cycle to fix their carbon. Actually, you have other microbes that can use the Calvin cycle too. Even some chemolithotrophs. A lot of chemolithotrophs actually are autotrophs, and a lot of them will be using the Calvin cycle to fix their carbon. You even have some archaea that are chemolithotrophic archaea that will use the Calvin cycle also to fix their carbon. But in addition to the Calvin cycle, you have 5 other autotrophic pathways known for carbon fixation. So the Calvin cycle is going to be taking in inorganic carbon dioxide and putting out glucose or some other carbohydrate. your other pathways for carbon fixation are all going to be taking in inorganic carbon dioxide and putting out acetyl-CoA. So they're changing those carbons in the CO2 to the carbons in the acetyl group on that acetyl-CoA, and then they can link those acetyl groups together to make whatever they need, whether it be sugars or lipids or nucleotides. They can make it from acetyl groups. And carbon fixation is very important because every organism needs carbon, of course. Carbon is the basis for all organic molecules. An organic molecule is actually defined as just a molecule based on carbon. So you need a lot of carbon to be able to grow and make all the molecules that your cells require. But you cannot get that carbon unless it's fixed. So the whole of chemoorganic trophic life is basically depending, or chemoorganoheterotrophic life is basically depending on carbon fixation. And most organisms are chemoorganoheterotrophs, organisms that are getting their carbon and their energy from organic molecules. That could not exist if you did not have carbon fixation. So first we'll focus on the Calvin cycle, and then we'll briefly go over a couple of the other pathways that can be used. The Calvin cycle would be occurring in the cytoplasm for prokaryotes, and in eukaryotes it would be occurring in the stroma of the chloroplast. So for eukaryotes, here's your chloroplast. These are the thylakoids, where you would be having your light reactions, and then this space outside of them, but inside the main membranes of the chloroplast, that's your stroma. So this is where the Calvin cycle will be occurring for your eukaryotes. The Calvin cycle is actually kind of complicated. There's a lot of different reactions in that cycle. And it's a cycle because the chemical that you start with is also the chemical that you end with by the time you get through the cycle. And that chemical is ribulose bisphosphate, or RuBP. So you start out with RuBP, you add some carbon dioxide to it, it goes through all these different forms, you end up making a sugar that you're able to take off the RuBP, and then, but it's not actually RuBP anymore, it's some other molecule that's similar to that. And then you have to regenerate your RuBP through another complex series of reactions until you actually have back that RuBP that you started with, and you can do the cycle again. The Calvin cycle is used generally to generate 6-carbon sugars, which are called hexoses. A glucose is an example of a hexose. It's the most common hexose. So they would usually be making glucose, but they could also make other sugars too. But actually, every time that you run the Calvin cycle, you only fix one atom of carbon. You only fix one molecule of carbon dioxide. So that means that if you're going to make a glucose that has 6 carbons in it, you have to run the Calvin cycle 6 different times. So in general, this is what organisms would be doing, running their Calvin cycle 6 times in a row to get out a single molecule of glucose. We won't talk too much about the details of how that happens, but it does need a lot of energy and a lot of electrons that are generally going to come from NADPH, but they could also come from NADH. So here's your overview of the Calvin cycle. Here's your RuBP that you start with. Then you're going to have carbon dioxide added in to the RuBP, and you have an enzyme that does that, which is ribulose bisphosphate carboxylase, or Rubisco for short. So you might have heard of Rubisco before. It's sometimes considered the most important enzyme that exists because it has this role in starting off the Calvin cycle, which is used to fix carbon, so by all eukaryotes and by many prokaryotes. And then, of course, that carbon is needed for all other forms of life that cannot fix carbon for themselves. So this Rubisco enzyme is going to take your RuBP and attach a molecule of carbon dioxide to it. Then that gives you some intermediate, which we're not going to worry about what it is exactly, but you're going to go through this cycle, going through different intermediates. You have different enzymes catalyzing each step, and you need a lot of ATP coming in here. You need a lot of electrons from your NADPH or NADH. Ultimately, you're going to get a molecule out that has 6 carbons in it if you have run this 6 times. So that molecule is going to be kind of waiting there until you go around 6 times and add 6 carbons to RuBP. Then eventually you will have enough carbons on this molecule, that's your output molecule, to actually make it a 6-carbon glucose. Overall, to run this cycle 6 times to get all the carbons you need for glucose, you're going to need to put in 12 electron carriers, 12 NADPHs or 12 NADHs, and you're going to need to put in 18 ATPs molecules. So that's very expensive. 12 NADHs or NADPHs and 18 ATP for a single glucose molecule. And this would be the reason why most of the products of your photophosphorylation are going just to the Calvin cycle or to whichever other cycle or pathway you're using to fix carbon. It's very expensive. Another pathway that's used is the reverse citric acid cycle. This would be used by your green sulfur bacteria. Also, it's used by some chemolithotrophic prokaryotes, both bacteria and archaea, although most of those chemolithotrophs would be using the Calvin cycle. The reverse citric acid cycle is really similar to the Krebs cycle or the citric acid cycle. It's basically the Krebs cycle backwards. Since you're going backwards through the Krebs cycle, you need to use a different set of enzymes though. So organisms that can do the citric acid cycle cannot necessarily do the reverse citric acid cycle. They would need to have the specific enzymes that they need for it. This is also a very expensive process, but it's cheaper than the Calvin cycle. So it's not requiring 18 ATP, it's requiring 10. Not requiring 12 NADPHs or NADHs, just requiring 4. But on the other hand, you're not making a whole glucose, you're making an acetyl-CoA. So you're fixing less carbon overall. You would be fixing, in the Calvin cycle for one glucose, 6 atoms of carbon. For the reverse citric acid cycle for one acetyl-CoA, you're only fixing 2 atoms of carbon. So here's your reverse citric acid cycle. The citric acid cycle always starts with oxaloacetate, and in this case you're going backwards, so instead of going this way, you would go this way. But you're also going to end back at oxaloacetate. So here you're actually adding electrons into your oxaloacetate first, and then ATP as well, and then you get into adding carbon dioxide. And you're going to add ultimately 2 different molecules of carbon dioxide to that oxaloacetate intermediate. And eventually you get citrate, and from there you can remove those couple extra carbons, add them to CoA to get acetyl-CoA, and then that ends up with you having oxaloacetate again. Then this acetyl-CoA can be used to make glucose or different sugars that you need, basically by doing pyruvate oxidation backwards and then doing glycolysis backwards, or that is doing gluconeogenesis. And we'll go over a couple of the other pathways that are used real briefly. First, you have the 3-hydroxypropionate bicycle. It's called a bicycle because it's actually 2 different half cycles that need to occur. We will not be talking about the details there really, but this is used by not very many organisms that live today. You have just some of the green non-sulfur bacteria that use it. They would be found in the genus Chloroflexus, and some chemolithotrophic archaea that use it. No one else uses it. We're only mentioning it because it's a very ancient pathway. The organisms that use it, the Chloroflexus genus bacteria and those chemolithotrophic archaea, they're some of the oldest organisms that we know of in terms of their phylogeny. So that means that they diverged right at the beginning when bacteria split off from archaea. Not long after that, you had Chloroflexus arise and these chemolithotrophic archaea. And they had this pathway, the 3-hydroxypropionate bicycle, which means that this pathway is very old as well. If the organisms that have it are very old, then it must be correspondingly old, which makes us think that it's maybe even the first pathway for carbon fixation. If it's not the first, it must be like almost the first. I don't know if you guys can hear this, an extreme amount of honking that's going on outside right now for some reason. It almost makes me wonder what's happening. But okay, so this 3-hydroxypropionate bicycle is the oldest that we know of pathway for carbon fixation, which would make it very important in the history of life. If it was the actual first carbon fixation cycle, then this would be basically what allowed heterotrophs to even exist in the first place. And the first organisms, which would have been probably autotrophs, would have been using this pathway to fix their carbon and would not have been able to exist without it. Then we have the reductive acetyl-CoA pathway, not to be confused with the reverse citric acid cycle. This is also commonly used by a number of different bacteria and archaea. Usually they're actually not necessarily phototrophs though. A lot of them are going to be methanogens or acetogens, which would be organisms that create methane or create acetate through the operation of their metabolism. So we'll actually mention these guys later on in the lecture or in the chapter. And we'll conclude this section by talking about nitrogen fixation. So nitrogen fixation is not autotrophy. Autotrophy means fixing carbon, or getting carbon from an inorganic source, which would be fixing carbon. Nitrogen fixation is doing the same thing but to nitrogen. So it is not autotrophy, but it is fixation. So instead of fixing inorganic carbon to be organic carbon, you are fixing inorganic nitrogen into not an organic form, but a form that is useful for organisms, ammonia for the most part. So you can define nitrogen fixation as reducing nitrogen or molecular nitrogen in the atmosphere to ammonia, which is NH3. So very few organisms are able to use atmospheric molecular nitrogen as a source of their nitrogen, even though every organism needs nitrogen to build their nucleotides and their amino acids. But nitrogen fixers can use molecular nitrogen from the atmosphere, and they will convert it to ammonia, NH3, which most organisms are able to use. Not all organisms, some organisms will not use ammonia, they will use nitrate instead, but most organisms will be able to use ammonia. There's a lot of different nitrogen fixers amongst the prokaryotes, the bacteria and their archaea. And they cover kind of a broad range. So some of them are phototrophs, some of them are chemoorganotrophs that would be getting their energy not from light but from chemicals, organic chemicals specifically, and some of them are chemolithotrophs that are getting that energy from inorganic chemicals. No matter what type of organism it is, all organisms that do nitrogen fixation use the nitrogenase enzyme complex to do it. The nitrogenase complex is actually responsible for reducing that molecular nitrogen to ammonia. The nitrogenase complex is interesting because it is irreversibly inactivated by oxygen. So if this complex is exposed to oxygen, you're going to have a reaction occur between the complex and the O2 that cannot go backwards. You cannot fix it. It will be inactivated forever after that. And the organism that has that complex would need to destroy it and make a new one, and hopefully there's no more oxygen around. So all of your nitrogen fixers have some way of protecting their nitrogenase complexes from oxygen. Different organisms will have different solutions. Some of them just don't live where there is oxygen and they're anaerobes. Some of them have a more intricate solution. In addition to needing the nitrogenase enzyme complex, nitrogen fixation also requires a good amount of energy and electrons. So it's requiring 16 ATP just to make 2 ammonia. You're going to make, for every N2 that you fix, you're going to make 2 different ammonias since there's 2 N's in there. And you also need 8 electrons, but those are not going to come from an electron carrier actually this time. They're just going to come from pyruvate. So the nitrogenase complex is going to strip those electrons off of pyruvate and add them to N2 along with ATP, also picking up some hydrogens, and that will give you NH3. The reason why this is so expensive is because molecular nitrogen we're into is a very stable molecule. It takes a lot of energy to break those bonds between the nitrogens so that you can turn each one into an ammonia. And on the side here you have that basic process of nitrogen fixation. First generating the electrons for nitrogenase, and then the nitrogenase actually using them to fix the nitrogen. Here's an overall summary of that happening. It's a little easier to kind of look at. But actually you don't need to know this process. You just know that we're taking molecular nitrogen and converting it to ammonia in a process that is requiring electrons and ATP. So now we'll start going through the different types of respiration. So you can categorize respiration based on the different electron donors that are used or by the different electron acceptors that are used. The electron donors are giving up their electrons at the beginning of the electron transport chain. The electron acceptors are accepting the electrons as they come off of the electron transport chain. First, we'll go through the different electron donors that can be used. The organisms that are using these different donors would include chemoorganotrophs that are using an organic chemical as that electron donor, and they would also include chemolithotrophs that are using some inorganic chemical as the electron donor. But we're mostly going to be focusing on the chemolithotrophs. For the chemoorganotrophs, we really went over the process in detail in Chapter 3 already. So first off, there's a lot of different donors and acceptors that can be used for respiration. Also a lot of different protein complexes that could be a part of that electron transport chain. Just a lot of different ways to get it done. If you're a chemoorganotroph, then you are taking electrons from an organic compound, which means that you are oxidizing that organic compound. So you're getting your carbon from there, but you're also getting your energy from there in basically the same process. If you're a chemolithotroph, then instead of using an organic compound, you use an inorganic compound, and you oxidize that compound by removing electrons from it that will enter the electron transport chain. Most of those chemolithotrophs have to get their energy and their carbon from a different place. So their energy is coming from respiration, using those inorganic compounds as electron donors, but their carbon is coming from somewhere else. A lot of them are autotrophs, and they're going to have some way of fixing carbon, either the Calvin cycle or the reverse citric acid cycle or something else. So for all those different types of respiration, there's a few things that we always have in common between all of them. You always are going to have an electron transport chain. You're always going to have an electron donor giving electrons to that chain and becoming oxidized in the process. And at the end of the chain, you will have an electron acceptor that will take the electrons from the chain and be reduced in the process. So when we say that an organism oxidizes some compound in respiration, we mean that that compound is the electron donor that they use for respiration. If we say that an organism reduces some compound in respiration, we mean that they're using that compound as the electron acceptor at the end of their electron transport chain. Since it's taking electrons, it's becoming reduced. In general, when your electron transport chain is operating and electrons are moving on it, you have hydrogen ions being pumped across the membrane to create a proton motive force. But sometimes it's different. Sometimes you don't pump protons, but you would pump some other ion, like maybe sodium. So you're not making a proton motive force, but maybe a sodium motive force or some other ion. Then your ATP synthase is going to be using that motive force, usually the proton motive force, to make ATP. And just as a refresher, we have a diagram of the basic oxidative phosphorylation process in respiration right here. So you're going to be having here your electron donor, which is NADH. Actually, This is not the first donor, as you remember, because this is not the only process that's part of respiration. This is just the oxidative phosphorylation part. So before you even get to here, you would have had your Krebs cycle, your pyruvate oxidation, and your glycolysis. Whichever molecule is starting there, that's the ultimate primary electron donor. The electrons are being stripped from that molecule, generally glucose, added to different electron carriers, and those electron carriers take the electrons to the electron transport chain and drop them off. So this would be the electron transport chain for maybe a heterotroph, a chemoorganoheterotroph that is using glucose for carbon and for energy. So it's stripping the electrons off of glucose in glycolysis and pyruvate oxidation and in the Krebs cycle, and those electrons are being added to electron carriers that take them to oxidative phosphorylation and the electron transport chain. Those electrons will then be dropped off on the electron transport chain, and they'll be transferred between the different proteins and electron carriers of the chain until they get to the end eventually. Then when they're at the end, they're going to be added to the final electron acceptor, which for this chain that's shown is oxygen, which makes water. So this would be aerobic respiration. In the process, hydrogen ions or protons are being pumped across the membrane at the different protein complexes of the electron transport chain, and that creates a proton motive force that would then be used by ATP synthase to make ATP. So before we go on to talk about the different electron donors that can be used for respiration, we'll talk about assimilation and dissimilation. You can have assimilative reductions, dissimilative reductions. You can also have assimilative oxidations and dissimilative oxidations. So this is something that is applying to specific molecules: nitrate, or NO3-, sulfate, or SO4-2-, and carbon dioxide, or CO2. All of these three molecules can be used in different ways by different organisms. So they could be used as the final electron acceptor in respiration, in anaerobic respiration. If you're an organism that is doing that, we're calling that dissimilative reduction. You are reducing those compounds by adding electrons to them, and that is dissimilative because you are using it for energy. You are not using it to build molecules in the cell. On the other hand, if you are an organism that's using nitrate, sulfate, or carbon dioxide, you're reducing those things, and it's not to get energy out of them, but instead it's to create your molecules. That is assimilative reduction. So dissimilative reduction is done for energy, so it's creating ATP. It's requiring electron carriers. You're going to be reducing that nitrate to molecular nitrogen, or that sulfate to hydrogen sulfide, or that carbon dioxide to methane, or some other similar product. So those are your waste products coming out of it. They're used as the final electron acceptor in your electron transport chain for respiration. So you're just adding electrons to them. Sometimes some hydrogens will come as well, and you get whatever product you get as a waste product, as well as your energy, your ATP. Not every microbe can do that, of course. It would only be specific anaerobic microbes that are using those compounds as electron acceptors in anaerobic respiration, which means that most microbes cannot do dissimilative reduction. For assimilative reduction, on the other hand, that's quite different. It does not generate energy. You're not doing it for energy, you're doing it to make the parts of the cell or the structures of the cell. So instead of making energy, this process requires energy. It also requires electron carriers. Instead of converting nitrate to molecular nitrogen, you would be converting it into an amino group that is added to some organic molecule like an amino acid. Instead of converting sulfate to hydrogen sulfide, you're converting sulfate to a sulfhydryl group or an SH group that would be added maybe to certain amino acids. Instead of reducing carbon dioxide to methane, you would be reducing carbon dioxide to part of the skeleton of an organic molecule, maybe a sugar, maybe a 6-carbon sugar, maybe an amino acid, maybe a nucleotide, whatever it is that you need. So that means that these are not waste products. These are added to a larger molecule to create a macromolecule for the cell. So assimilative reduction is not for energy, it's for making components of the cell. It's a type of anabolism because you're building those molecules, the nitrate, the sulfate, the carbon dioxide, into a bigger molecule. On the other hand, dissimilative reduction is catabolism because you are breaking those molecules down to get energy out of them. And the final difference is that pretty much every organism can do assimilative reduction, at least one type of assimilative reduction, because they need this to get, their stuff. They need it to build their molecules. So not every organism, of course, can use CO2 as a source of carbon. Only autotrophs can. But if you're not able to use CO2 as a source of carbon, you're probably able to either use nitrate as a source of nitrogen or sulfate as a source of sulfur. Whereas only some microbes can do dissimilative reduction. So in general here, we're going to be interested in dissimilative reducers who are using those compounds for respiration to get energy. We're not going to be interested in assimilative reducers, which are very common. Everybody basically can do one type of that or another, and they're all over the place. Okay, so first, the first electron donor that you could be using for your electron transport chain and respiration is molecular hydrogen, or H2, which would mean that you are oxidizing that hydrogen, oxidizing that molecular hydrogen by stripping electrons off of that and sending them onto an electron transport chain. If you are somebody who's able to do this, you are a chemolithotrope. A chemolithotroph, that is. Right, you're not using an organic molecule as a source of electrons for your electron transport chain. You're using molecular hydrogen, which is an inorganic molecule, so you're a chemolithotroph. Usually these are aerobes as well, which means that the final electron acceptor that they're using is oxygen. You also have some of these guys that are facultative chemolithotrophs, which means that if there's glucose or other organic molecules around that they could use, they're going to use those for respiration as electron donors. And they're going to be basically living a chemoorganoheterotrophic lifestyle. But when those glucose molecules dry up and run out and there's molecular hydrogen around, they're going to switch to using molecular hydrogen and living a chemolithotrophic lifestyle. In order to do this, to use hydrogen as an electron donor for respiration, you have to have the enzyme hydrogenase. So that enzyme is going to be found in the membrane of these molecules, and it's going to be in the membrane of these microbes, that is, and it's going to be used to strip the electrons off of the hydrogen, of the molecular hydrogen. Once those electrons have been removed, what you have left is protons, hydrogen ions, or just protons. So here you have an example of an electron transport chain using hydrogen as the electron donor. So here's your hydrogen coming in. Here's the hydrogenase enzyme, which is going to strip electrons off of the hydrogen, and then you get protons as a result. Those electrons travel on the electron transport chain. They end up being added to oxygen to make water. As they travel, hydrogen ions are pumped. Of course, you're creating a proton motive force partly just by stripping protons off of hydrogen on the outside of the cell. You're also pumping some more protons, and ultimately that gives you your proton motive force that you then use to power the ATP synthase and make ATP. These organisms are also able to get electron carriers out of this process. There's actually a couple of different ways that they can do that, which we're not really going to worry about, but they are able to use some of that hydrogen to make to charge their electron carriers with electrons, and they can use those electron carriers then for maybe for fixing carbon by an autotrophic pathway like the Calvin cycle, which would be assimilative because it's not done for energy, it's done to make molecules for the cell. Next you have sulfur compounds. There's a number of different sulfur compounds that can be used by different microbes as a source of electrons for their electron transport chains. Usually the microbes that are doing this are archaea, or they are colorless sulfur bacteria. So the colorless sulfur bacteria are different from green sulfur bacteria and purple sulfur bacteria. Those guys are phototrophs and they do not do this. The colorless sulfur bacteria don't have a pigment. They don't do photosynthesis. Instead, they're using sulfur compounds as electron donors for respiration. Most of the microbes that are doing this are aerobes, but you also have some of them that are anaerobes. So most of them would be using oxygen as their final electron acceptor. Some of them would not be doing that. Most of those guys would be using nitrate instead, which also note would be a form of dissimilative nitrate reduction. And usually these guys are autotrophs that would use the Calvin cycle to fix carbon. So there's a lot of different sulfur compounds that could be used as electron donors for respiration. They would include hydrogen sulfide or H2S, just S itself, so pure elemental sulfur, also thiosulfate, which is S2O32-, or sulfite, which is SO32-. No matter which product they're using, or no matter which compound they're using as a donor, they're going to be reducing that, oxidizing that donor to sulfate, SO42-, and to hydrogen ions. So they're going to be separating some hydrogen ions from this, or just protons, from whatever electron donor they're using. So because one of the products of this process is protons or hydrogen ions, that means that organisms that do this are generally manufacturing a more acidic environment for themselves. So they're going to need to be able to deal with that. So most of them are either going to be acidophilic or acid tolerant at least. Otherwise, they need to have some more complicated way of handling the low pH that they are manufacturing by their metabolism. There's a lot of different ways to do this. This is kind of one of the most common ones. We're not going to worry about the details of this, but you're starting with, in this case, elemental sulfur, which is not soluble. It's going to be just a little bit of a little bit of rock, and they're going to have to leach sulfur off of that rock. So once they've leached the sulfur off of the rock, it's going to be transported into the cell. Electrons will be stripped from it and will enter the electron transport chain. Ultimately, that's going to give you sulfate. Those electrons are going to move on the electron transport chain until the end, where they're added usually to oxygen, but maybe to nitrate instead, as the final electron acceptor. As they're moving, you're going to have protons that are being pumped from the inside of the cell to the outside of the cell. So some of the protons that are made when you oxidize whichever sulfur compound this is, in this case elemental sulfur, to sulfate, some of those protons are just going to be pumped right back out of the cell to the outside environment, creating the proton motive force, which then is used by ATP synthase to make ATP. Some microbes are able to use iron as their electron donor for respiration, which would be iron oxidation. And these are the iron bacteria. These are all chemolithotrophs, and they're generally aerobes, so using oxygen as their final electron acceptor. Most of them are also autotrophs that use the Calvin cycle to fix carbon. Some of them are anaerobes instead of aerobes that would be using nitrate usually as their final electron acceptor. Which would mean that they are dissimilative nitrate reducers as well. And a few of them are actually phototrophs that are oxidizing iron in order to get electrons for the Calvin cycle. So that would be a bit different from oxidizing iron as an electron donor. Right? If you're oxidizing iron as an electron donor, that would be more like dissimilative. If you're oxidizing it in order to run the Calvin cycle, then that's more like assimilative. Usually these guys are also acidophilic because their metabolism will also produce protons. So when you oxidize iron, you're generally going to be oxidizing the Fe2+ ion. Then when you strip an electron off of that, what you get is the Fe3+ iron. So Fe2+ iron is ferrous iron, Fe3+ iron is ferric iron. So these guys are going to be oxidizing ferrous iron to ferric iron. When they do that and they make that ferric iron, if there's any water around, it's going to immediately react with the water in a process that creates protons. So that means that as they're running their metabolism, creating ferric iron, they're going to be also creating protons as well, unless they're in an extremely dry environment, which they would not be in general. Also, if they were not at an acidic pH due to all those extra protons, they would have a problem, because if you're at a neutral pH or an alkaline pH, ferrous iron is actually going to spontaneously oxidize to ferric iron. And if all your ferrous iron is just spontaneously oxidizing to ferric iron, then you're not able to strip electrons from it and use those electrons for your metabolism. So that's no good. So these guys are generally going to be acidophilic, at least acid tolerant, but usually acidophilic, and actually some of them are extremely acidophilic. Some of these iron bacteria are the most acidophilic bacteria that are known. So because they live in such an acidic environment, usually with a lot of extra protons around, they generally do not need to use their electron transport chain to pump protons across the cell membrane and create a proton motive force. There basically already is a proton motive force because there's so many protons outside of the cell. Because whenever they're oxidizing their electron donor iron to ferric iron, that's going to stay outside of the cell. So it's going to react with water outside of the cell and create protons outside of the cell. So just by doing that, they're already making basically a proton motive force. So that's not a need for them to create using the electron transport chain. Instead, they're using their electron transport chain to take protons out of their cytosol, which gives them a more neutral internal pH. So that's part of how they're kind of surviving in this deeply acidic environment, is by consuming protons from their cytosol, which when you have less protons, the pH will go up, so the pH will go up to a more neutral pH that is like livable. So here you have an example of that. This is a gram-negative iron bacteria that's shown. So here in the outer membrane, they would actually have a protein complex that is designed to strip electrons off of ferrous iron, oxidizing it to ferric iron, and then you're going to have that reacting with water to make protons. Then those electrons that were taken from the ferrous iron are going to be moved through the periplasm to an electron transport chain inside the cell. Some of those electrons are actually going to go to make NADH, which they would then use for the Calvin cycle. Some of those electrons are going to go to the rest of the electron transport chain and be added to oxygen, usually as the final electron acceptor, making water. In that process, you are taking some protons and pumping them to the outside just to remove them from the inside so that you can have a more neutral pH in here. Also, whenever you add those electrons to oxygen to make water, that requires some protons as well. So you have your oxygen here, but H2O is water, so you need hydrogens. You're getting electrons, that's half a hydrogen already, but you need protons to make up the rest of the hydrogen. So oxygen plus an electron plus a hydrogen would mean oxygen with an H attached to it, and if you get two of those H's, that's H2O. So they're consuming protons from their internal environment when they drop their electrons off onto oxygen, making water. And then, of course, they're able to use those hydrogen ions or those protons from outside of the cell to run their ATP synthase and make ATP. In general, this is not an efficient way to make energy. You have to oxidize a lot of iron to consume enough protons from the inside of the cell to maintain your PMF and make ATP this way. So it's not the best way to do it, but it is a way that they're able to get by in environments that have a lot of iron. So they're going to be creating a lot of waste by running this process and not making a lot of ATP. Next we have nitrification, which is the oxidation of ammonia or nitrite. Ammonia is NH3, nitrite is NO2-. So nitrification as a whole is actually oxidizing ammonia, NH3, to nitrate, NO3-, but it actually has two parts. You can't do that just in one step in general. You have to do it in two different steps. So first you're oxidizing your ammonia to nitrite, NO2-, then you can oxidize nitrite to nitrate, NO3-. Actually, most organisms that do this will not be able to do both parts. They will do just one or the other oxidation. Any microbe that is able to do either one of those parts, either oxidizing ammonia to nitrite or oxidizing nitrite to nitrate, those are all going to be called nitrifying bacteria or nitrifying archaea. They're all going to be chemolithotrophs. Generally, they're aerobes, so they're using oxygen as their final electron acceptor. Most of them are autotrophs that use the Calvin cycle to fix CO2. And then a lot of them are also going to be facultative chemolithotrophs, which means that when they're able to, they're going to live off of organic molecules like glucose, and then when those are not available, then they would switch to the nitrification process, oxidizing ammonia or nitrite as an electron donor. So if you're an ammonia oxidizer, that means that you are a nitrifying bacteria or archaea that does the ammonia oxidation, oxidizing ammonia to nitrite. If you're a nitrite oxidizer, then you are oxidizing nitrite to nitrate, using nitrite as your electron donor for respiration, for aerobic respiration. There actually are just a few nitrifying bacteria that are able to do both processes, both the ammonia oxidation and the nitrite oxidation, but almost all microbes who do this are only going to do one or the other. They're going to fall into one group or the other group. Here you have a diagram of those processes. So here you're starting off, at the top you have ammonia oxidation. You're starting off with ammonia here, which would be inside of the cell. And you're reacting it, we're not going to worry about the details here really, but you're reacting it with oxygen and protons in a process that strips an electron off of that ammonia. So that stripped electron is going to end up on the electron transport chain. Meanwhile, this ammonia is going to be converted ultimately to nitrite, NO2-, through kind of a little chain of reactions, and it's also being transported outside of the cell. Your electrons from that ammonia that ended up on the electron transport chain will travel through the electron transport chain. Some of them are actually going to leave and be added to an electron carrier like NAD+, making NADH. Those electrons are going out to fuel the Calvin cycle. The rest of those electrons will continue on the electron transport chain to the end, where they're added to oxygen to make water. And then in the process, you are also pumping protons across the membrane, creating a proton motive force that's used by ATP synthase to make ATP. So that's for ammonia oxidizers. If you are a nitrite oxidizer, then you are transporting that nitrite into the cell, and you're reacting it with water to make nitrate. In that process, you are stripping an electron off of the nitrite, so you're oxidizing it. That electron will enter the electron transport chain. Some of those electrons will actually be added to NAD+, making NADH, again used to fuel the Calvin cycle. The rest of those electrons are going to continue on the electron transport chain to the end, where they're added to oxygen to make water. And in that process, you are pumping protons across the membrane, making a proton motive force to run ATP synthase. So similar to iron oxidation, this is not an efficient way to make your ATP. You have to go through a lot of ammonia or a lot of nitrite to make enough ATP to fuel the cell, which would be why these guys like to live off of glucose when it's available. And it generates a lot of waste. But in this case, the waste, it's either nitrite or it's nitrate. It's not exactly like a bad thing for for the organisms that are around these nitrifying bacteria. Usually these guys like to live together, so you'll have some of them there oxidizing ammonia to nitrite, then some other guys oxidizing nitrite to nitrate. So they're manufacturing nitrate as a whole. That nitrate is going to be useful for other organisms in the environment, especially plants. course, you have other microbes that can use nitrate as a final electron acceptor for their electron transport chain, but plants are going to need a lot of nitrate. So these guys are really important for those plants. Also, they're useful in treating wastewater because they can remove ammonia from the wastewater. Next, we have Anammox, which is anaerobic ammonia oxidation. So this is another form of ammonia oxidation, but it is not part of nitrification. The difference is that in nitrification, ultimately you're making nitrite, you're oxidizing that ammonia to nitrite, and then a nitrite oxidizer will oxidize it to nitrate. In Anammox, you are oxidizing ammonia to molecular nitrogen. So Anammox is ammonia to molecular nitrogen. Nitrification is ammonia to nitrite and then to nitrate. Anammox is a process that's known in different bacteria. Those bacteria that are Anammox bacteria are also autotrophs that will use the reductive acetyl-CoA pathway to fix their carbon. These are also guys that have an actual organelle, which is very rare for prokaryotes to have organelles, but Anammox bacteria will have one, the Anammoxosome, where they are doing the Anammox process. They have to do it in a special organelle. They can't just do it in the cytosol because the intermediates of this process are quite toxic. In addition to requiring ammonia for Anammox, you also actually need nitrite too. And we're not going to really worry about these details, but here's the diagram. So this would be the membrane of the Anammoxosome. This is the cytosol of the cell, and then this is the interior of the Anammoxosome. So you're going to be transporting ammonia and nitrite into your Anammoxosome if you're one of these Anammox bacteria. The ammonia is actually going to end up converted to ammonium ion because the pH is quite low in here. And it's going to be entering this process where you are ultimately stripping electrons off of it, stripping protons off of it as well that stay back inside of the Anammoxosome. Those electrons will enter an electron transport chain. They're going to come out the other end of that chain and be added to nitrite, and ultimately that process feeds back into the ammonia. You're stripping more electrons off of that ammonia, and this is kind of a cycle. It's going to go in a little cycle here. Those protons that you're pumping into the Anammoxosome by the operation of the electron transport chain will then be used to run ATP synthase and make ATP. So this process is making ATP, but it actually does not make electron carriers. Since these guys are autotrophs, they're going to need electron carriers for their reductive acetyl-CoA pathway, but they're not able to get those electron carriers from the Anammox process. They can only get ATP from here. So they have another process where they're going to oxidize nitrite and use the electrons to make NADH for carbon fixation. And a final point about the Anammox bacteria is that they like to live in conjunction with the nitrifying bacteria that are oxidizing ammonia to nitrite and then to nitrate. This is a good way for them to get nitrite. If they're living in a place that has a lot of ammonia, some of those nitrifying bacteria will oxidize it to nitrite for them, and they can use that. Also, these guys are useful for wastewater treatment. Since the final product here is molecular nitrogen, which should be shown right here actually, the molecular nitrogen, that's actually a gas. So these guys are converting ammonia to a gas, molecular nitrogen. So it's a really useful way to remove ammonia from wastewater, because since the product is a gas, it'll just kind of leave the water. Okay, so those were our major electron donors for respiration. Not all of them, just the major ones. And now we'll go through the major electron acceptors for respiration. So everybody who's using these different acceptors is going to be some type of anaerobe, because we're not going to mention oxygen here. So first we have denitrification, which is the process of using nitrate as a final electron acceptor, reducing it to form some type of gas, either nitric oxide, which is NO, or nitrous oxide, which is N2O, or elemental nitrogen N2. So it's called denitrification because you're taking nitrogen that's present in the soil or the water, wherever you're living, in the form of nitrate, and you're converting it to a gas. That gas is just going to leave and enter the air. So you are actually removing nitrogen from the soil or the water where you're living. You also have other microbes that can use nitrate as a final electron acceptor, but don't reduce it to a gas. They would reduce it to something else, maybe ammonia. So those guys are not doing denitrification because their product is not a gas. It's just going to stay in the soil or the water wherever they're living, and the total amount of nitrogen in that environment will not change. A lot of denitrifiers have other kind of tricks in their toolkit as well. A lot of them are facultative aerobes, so they can use oxygen as their final electron acceptor if it's present. Some of them that are not aerobes have to use something that's not oxygen as their final electron acceptor, but they can usually use something other than just nitrate. So maybe nitrate or a sulfur compound, for instance. So they have many kind of ways to get the job done. And this process is very important for the environment for a couple of different reasons, generally not for good reasons. So if these denitrifiers are producing nitric oxide or NO, that's going to go up into the atmosphere until it hits the ozone layer, and it's going to react with that ozone to form nitrite, and then that nitrite is acidic and it falls down in the rain to the earth, and that's basically one of the main components of acid rain. Then if they're forming nitrous oxide or N2O, that goes up into the atmosphere and it just stays there and absorbs light, or I should say reflects light back down to the earth. So nitrous oxide is actually a greenhouse gas that contributes to climate change. So those are the bad things about your denitrifiers, but there's also a good thing, which is that they're useful for wastewater treatment. Because they are taking nitrate in that wastewater and converting it to some type of gas that's just going to leave the wastewater. Of course, it would be great to then catch that gas because it's probably something like nitric oxide or nitrous oxide, which is poisonous for the environment. So here's the electron transport chain for denitrifiers, and we are not going to worry about these details once again, but you're starting with your electron donor. In this case, we show that as NADH, but actually those electrons would have been coming from somewhere else first, probably from an organic compound like glucose. So their ultimate electron donor would probably be something like glucose. Then those electrons from glucose get added to NADH and dropped off on the electron transport chain. So they're going to start traveling on the electron transport chain. As they move, protons will be pumped across the membrane, creating a proton motive force that is later used by ATP synthase to make ATP. Then there's several points at which these electrons can exit the electron transport chain. So they could exit right here and be added to nitrate, which forms NO2-. And then that NO2- is going to come down and be reacted to nitric oxide by this enzyme here, NO2- reductase. That process, converting NO2- to NO, also requires more electrons. So some of your electrons from the electron transport chain will travel down to this point and exit here to be added to NO2-, making nitric oxide. Some of them are going to continue on and leave the electron transport chain right here and be added to that nitric oxide, which forms nitrous oxide. Some of them will make it all the way to the very end and be used by N2O reductase to convert nitrous oxide to elemental nitrogen gas. So these guys are going to give off a combination of all of those gases. Next we have sulfate reduction and sulfur reduction, which are two similar things, but they're a little bit different. Sulfate reducing bacteria are going to be using sulfate as their final electron acceptor, and they reduce it to hydrogen sulfide. These guys are going to be obligate anaerobes that cannot tolerate oxygen. In general, they're also going to be chemolithotrophs that are using hydrogen as their electron donor. A few of them are also autotrophs that are going to be using the reverse acetyl-CoA pathway to fix their carbon. Then you have sulfur reducers that are able to use sulfur as their final electron acceptor, but they cannot use sulfate. So actually, for the sulfate reducing bacteria, most of them would be able to use sulfur as well as a final electron acceptor. So they can do both sulfate and sulfur in general, in both cases reducing it to hydrogen sulfide. The sulfur reducers, which include both bacteria and archaea, are also reducing sulfur to hydrogen sulfide, but they cannot use sulfate as their final electron acceptor. It must be sulfur, elemental sulfur. So that's like kind of a fine distinction, but that's the distinction between them. These guys are also going to be generally using hydrogen, molecular hydrogen, as their electron donor, or some organic compound. So they could be a chemoorganotroph, or they could be a chemolithotroph. Some of them will actually be able to switch and use organic compounds sometimes and molecular hydrogen at other times if organics are not available. So here's the basic overview for that process, which again, we are not going to worry about too much at all, really, in a way. So here's your start of the electron transport chain with molecular hydrogen. So you're going to have electrons being stripped off of that you're going to have electrons being stripped off of that molecular hydrogen. They're going to enter the electron transport chain and be transported through it. Ultimately, they're going to be added to, in this case, sulfate, because this is an example for a sulfate-reducing bacterium. Then that sulfate is going to be reacted to ultimately form hydrogen sulfide. So they're making a proton motive force in kind of an oblique way. This hydrogen, this molecular hydrogen that they're using as an electron donor is actually outside of the cell, and when you strip electrons off of it, you're left with protons. So that's your proton motive force right there. And then that proton motive force would be used by ATP synthase to make ATP. So the point of running that electron transport chain then would be to make electron carriers that they would be able to use for other things, such as, for some of them, fixing carbon. For the ones that are going to be able to use organic molecules as electron donors as well, they would do it in kind of a process like this, where they're ultimately reacting those organic molecules in a way that yields molecular hydrogen. So they're stripping molecular hydrogen off of those organic molecules, and then that molecular hydrogen is what is actually being used for the electron donor. So finally, we'll conclude our survey of respiration looking at some other final electron acceptors that can be used relatively commonly. So this is not an exhaustive list, of course. But you have some microbes that can use metals or metalloids as a final electron acceptor. A metalloid is an element that has properties that are kind of in between the properties of metals and nonmetals. Out of those, the most common to be used as a final electron acceptor would be ferric iron, Fe3+, which would be reduced to Fe2+, and manganese 4+, which would be reduced to manganese 2+. You can also find microbes that use selenium, tellurium, arsenic, or chlorine compounds as well. And a lot of these microbes are able to use multiple metals or metalloids as their final electron acceptor. So they're pretty flexible in terms of what they can use. Some of these guys are really useful for bioremediation, or the cleaning of pollutants in the environment using organisms, especially microorganisms. So you can use some of these microbes to clean up arsenic poisoning. A lot of those chlorine compounds can be quite toxic as well and can be detoxified using these microbes. Then you have a range of organic compounds that are used as final electron acceptors by different microbes. That would include fumarate, compound called, or abbreviated TMAO, which comes from fish and actually is responsible for the fishy smell when it is reduced to TMA by microbes. Also DMSO, which is actually a solvent that's commonly used in the lab, could be used as a final electron acceptor by a number of different microbes. And you also have some microbes who can use chlorinated organic compounds. An example of a chlorinated organic compound is the PCB, or polychlorinated biphenyl, which are common pollutants. So microbes that are using PCBs as a final electron acceptor are very useful in bioremediation as well. That process is called reductive dechlorination, or reductive dechlorination, sorry, where they are using the PCB to reduce it as a final electron acceptor, and in that process it loses the chlorine or the chloride atom attached to it and is detoxified. Finally, it's very rare, but there are some microbes, specifically some archaea, that are able to use protons as a final electron acceptor, which would generate hydrogen. So this is just kind of a couple of graphics showing bioremediation. So generally, if you have pollutants spill into the ground, you're going to have different zones where different types of microbes are doing different types of metabolism to detoxify or just use all of these pollutants that have been spilled into the ground. So you'll have a zone where microbes are doing methanogenesis and generating methane from their metabolism. You'll have zones where they're maybe reducing sulfates, reducing iron, reducing manganese, reducing nitrate. So different types of compounds would be detoxified in different zones by different microbes. And you can actually inject microbes into these different zones and inject the other metabolites that they need in order to perform their metabolism and clean up the pollutants. So this is an example of that actually, where you have some pollutants going down into the groundwater, and then as part of the bioremediation, they're actually injecting organics or carbon dioxide so that those microbes can use that for their metabolism and use a chlorinated compound that's one of the pollutants as their final electron acceptor, dechlorinating it. You also have PCBs that are found contaminating sediments at the bottom of the ocean or large lakes, and you can have microbes there bioremediating that as well by the same mechanism. So next we'll go over one-carbon metabolism briefly. So a one-carbon metabolism refers to processes in which organisms can use compounds that only contain one carbon for energy. So that would be things like carbon dioxide or methane, other compounds too, but usually those two. And usually this is anaerobic, so it does not require oxygen. Frequently, actually, the one-carbon metabolism does not involve an electron transport chain, and it uses enzymes that are kind of unique to it, which is why it's considered separately. So for organisms that are using carbon dioxide as their electron acceptor in a one-carbon metabolism, you can divide them into two groups, the acetogens and the methanogens. Acetogens generate acetate from carbon dioxide, methanogens generate methane. Both of those are autotrophs that are using the reductive acetyl-CoA pathway to fix their carbon. The difference is that acetogens would be using that pathway also for energy as well as to fix carbon dioxide, whereas methanogens would only use it to fix carbon dioxide. Some acetogens or methanogens will actually use a sodium motive force instead of a proton motive force. So even though this type of metabolism a lot of the time would not involve an electron transport chain, it still does involve pumping ions across a membrane to create an ion gradient, usually the proton motive force, and then that's used to make ATP. But sometimes also a sodium motive force would be used instead if they're pumping sodium, and then that can be used to make ATP by ATP synthase. by a special type of ATP synthase. In general, for these organisms, the electron donor that they're using would be molecular hydrogen, or it could also be another one-carbon compound like methanol. And they are always doing this in an anaerobic fashion. So in the diagram here, this is showing the metabolism for an acetogen, and we are not going to worry about these details, but you can see they're starting with carbon dioxide. They're converting that ultimately to acetate. They also would use another carbon dioxide, so they technically need two different carbon dioxides in order to generate one acetate, since that is a two carbon molecule. They're able to make a little bit of ATP in that process, and it requires a lot of electrons. They're also able to use some of their electron carriers to pump sodium across their membrane, and then that is used to manufacture ATP as a sodium motive force. And then this diagram here is just showing you kind of a comparison of methanogens versus acetogens. Both of them are starting with carbon dioxide. This is actually bicarbonate that's shown. HCO3- is bicarbonate, but anytime you put carbon dioxide in water, it will automatically react to form bicarbonate and a free proton. So this is going to happen no matter what, anytime carbon dioxide is added to water, which means that ultimately they are using carbon dioxide there. They are reducing it, adding electrons to it. The methanogens are generating methane that way, and the acetogens are generating acetate that way, and would require two carbon dioxides and not just one to make one molecule of acetate. In addition to this, they're also pumping ions to create a proton motive force or sodium motive force, which they're using to make ATP. Then we have the methylotrophs. Methylotrophs are organisms with a one-carbon metabolism that are going to use organic compounds as a source of energy and carbon, specifically organic compounds that do not have carbon-carbon bonds. So any organic compound that doesn't have a carbon bonded to another carbon is going to be an organic compound that only has one carbon. Every organic compound is basically made of a skeleton of carbon. So if you don't have any carbon-carbon bonds, that means there's only one carbon there. Usually those compounds they'd be using would be methane or methanol, which is CH3OH. And they're using that as both a source of energy and a source of carbon, which means these guys count as autotrophs. There's also a subtype of methylotrophs, which is the methanotroph. A methanotroph is a microbe that is using CH4 or methane as their source for energy and for carbon. Methylotrophs in general would use any organic compound without carbon-carbon bonds, usually methane or methanol. So a methanotroph is a methylotroph, but not all methylotrophs are methanotrophs. They're going to be using that compound as an electron donor. And they could use it in a fashion that requires oxygen or does not require oxygen. What you have shown there in the diagram is actually the methanotroph metabolism, and it does involve an electron transport chain. So here you have methane, which they are oxidizing as their electron donor. That would generally generate methanol, and then they're going to convert that methanol to CH2O, which is formaldehyde. That formaldehyde they're actually going to use as their carbon source. So they have a couple different ways to turn that into organic compounds and build like sugars and their different macromolecules from it. The electrons that are taken off of this methane as it's converted to methanol are entering the electron transport chain. They travel on that electron transport chain. In this case, they're being added to oxygen as the final electron acceptor to generate water, but you could also have examples where they are anaerobes and they would have something else as their final electron acceptor. As those electrons are traveling on the electron transport chain, the energy of their movement is used to pump protons across the membrane, establishing a proton motive force, and then ATP synthase uses that to generate ATP. So now we'll briefly go over some of the most common fermentations classified by their products. So first, a basic overview of fermentation. Fermentation is making ATP in a process called substrate level phosphorylation, which we actually haven't talked about before. So we've talked about making ATP in oxidative phosphorylation and respiration, and in photophosphorylation and photosynthesis. Both of those ways of making ATP are using an electron transport chain and an ion concentration gradient like the proton motive force, as well as ATP synthase to make the ATP. In substrate level phosphorylation, it's a lot simpler. You're basically just transferring a phosphate group from some energy-rich compound to ADP directly, and then that creates ATP. It has to be a compound that has enough energy when you break the bond, so you're removing that phosphate from it, that when the energy is released from that broken bond, it's enough to actually cause that phosphate group to attach to the ADP, giving you ATP. So we've actually seen this before in respiration, we just didn't call it out by name. So we mentioned that in glycolysis, you're making a little bit of ATP. Also in the Krebs cycle or the citric acid cycle, you make some ATP. Both of those are examples of substrate level phosphorylation, where you're just transferring a phosphate group from one compound to ADP. So in fermentation, you're making your ATP by substrate level phosphorylation. You're also going to end up making NADH. So whichever substrate you're using for your substrate level phosphorylation, you're going to be taking electrons from that and putting them on NADH. So you're reducing NAD plus to NADH while oxidizing this substrate, and that process will also generate ATP. It could be glycolysis. It usually is glycolysis, but it could also be a different process that is also generating ATP and NADH by substrate level phosphorylation. Then you need to turn over your NADH. You need to oxidize that back to NAD+, so you can do another round of this process that is making your ATP. So in the process of removing those electrons back off the NADH and adding them to a fermentation product, you're going to be generating a fermentation product as some kind of waste. So usually organisms would be using NADH as their electron carrier for fermentation, but sometimes they might use something else. A lot of the time you'll see pyridoxine, the pyridoxine protein used, or it could be other things as well. For the cell that is doing this fermentation, the fermentation product that it ultimately makes is considered a waste product. So it's going to excrete it from the cell. But that doesn't mean that it's not useful to other organisms that are living nearby. So sometimes you'll have somebody's fermentation product serving as someone else's substrate, and they'll just take it right up and use it as an electron donor for their own fermentation reaction, or even for respiration. You can classify fermentations in a couple of different ways. You can classify them either by the substrate that's used, so that's that energy-rich compound that is fueling your substrate level phosphorylation, or you could classify them by your fermentation product. So the ones that we're going to look at are classified by fermentation product. And in the diagram here, it's kind of summarizing the whole process of fermentation. So you're taking in some organic compound, which might be your energy-rich compound, or maybe you have to convert it to the energy-rich compound before you can use it in substrate level phosphorylation. In that process, you're going to be oxidizing this compound, transferring some electrons to NADH or another electron carrier. Then this energy-rich compound is going to be used to create ATP by substrate level phosphorylation and ends up as an oxidized compound. Then you're going to convert that to your fermentation product by reducing it using the electrons from NADH, which gives you back NAD+. Then that fermentation product is waste, so it is excreted from the cell. So one of the most important types of fermentations is lactic acid fermentation. It's also very common. Lactic acid fermentation is any type of fermentation that produces lactic acid as a fermentation product. The bacteria that do this type of fermentation are just called lactic acid bacteria. For a substrate, they could use different things, but they're generally going to be using some type of sugar, which means they might be using glycolysis as their pathway for substrate level phosphorylation. You could divide lactic acid bacteria into two types, the ones that do homofermentative lactic acid fermentation and the ones that do heterofermentative lactic acid fermentation. So the homofermenters are producing only lactic acid from their fermentation. They're using glycolysis to make ATP, then to complete the fermentation they're producing lactic acid. The heterofermenters are producing lactic acid and other fermentation products as well. It's just that the biggest one is the lactic acid. So these guys are not going to be using glycolysis to make ATP. They would use a different pathway. It looks actually kind of similar to the pentose phosphate pathway, at least at first. They're going to be making not as much ATP by that pathway as you can make in glycolysis, and in addition to generating the lactic acid fermentation product, they would be generating ethanol and carbon dioxide as well. So here you have the process for homofermentation of glucose to give lactic acid. So at first we just have glycolysis. This is all just glycolysis. Actually, this whole thing is glycolysis until you get to the very end when you are converting pyruvate to lactate. So in this glycolysis, you're first, of course, using ATP, then at the end of it, you're making ATP, and at some point, you're also transferring electrons to NAD+, giving you NADH. Then once you have produced pyruvate from glycolysis, you're going to use that and reducing it basically, using those electrons from NADH, transferring them to pyruvate to reduce it to your fermentation product of lactate, which gives you back NAD+ and lets you do more glycolysis. And here's the pathway for heterofermentation that produces lactic acid. We're not really going to go over this, but you can see it's also producing ethanol here and carbon dioxide right here. It's also making not as much ATP. It's using 1 and making 2 rather than using 2 and making 4. Then you have mixed acid fermentations, which are a series of fermentation reactions in which your fermentation product is some type of acid, usually multiple acids. Could be acetic acid, which is vinegar, lactic acid, or succinic acid, or it could be some other type of acid as well. Those are just kind of the most common ones. And you actually have bacteria that are going to make all three of those in their mixed acid fermentation. These are generally going to be heterofermentative reactions where you're also generating other products, including ethanol, carbon dioxide, and molecular hydrogen. The substrate that's used is always going to be some type of sugar. So that means if you're doing glucose, you might be using glycolysis as the first part of the fermentation. And in general, you're going to see this type of fermentation performed by bacteria that live in intestines of animals. You also have a special subtype of mixed acid fermentations, which is the butanediol fermentation, in which the main product is butanediol. So this is that butanediol fermentation, starting with glucose, then you're doing glycolysis to make pyruvate, which is also where you're making your ATP and your NADH. Then this pyruvate is converted to butanediol, ultimately, in a process that is consuming that NADH that you created in glycolysis right there. Here's another type of, a more common type of the mixed acid fermentation where you're starting out with glucose, you're doing glycolysis on it, and you're generating a number of different fermentation products. So here they're generating acetate, ethanol, molecular hydrogen, carbon dioxide, lactate, and succinate all at the same time. So you can have lactic acid made as part of lactic acid fermentation or as part of a mixed acid fermentation. But the difference is that when lactate or lactic acid is produced in a mixed acid fermentation, it's also being produced with other acids as well. In lactic acid fermentation, the lactic acid is the only acid produced, except for carbon dioxide, which is technically a weak acid as well. One bacterial genus called Clostridium contains species that do clostridial fermentations. These are organisms that have to get all of their ATP from fermentation. So they don't have any type of respiration, any type of electron transport chain. It's all fermentation as far as the source of their energy. You have different species in that genus that can do fermentation using different substrates. Some of them are going to use sugars, some of them would use amino acids, some of them are going to use nucleotides. So they're very diverse in terms of the different things they can use. And you're actually going to find this type of bacteria infecting wounds sometimes, and eating all of the different products that are there. So all of the macromolecules in your body, it can basically, except for lipids, the Clostridia cells could ferment, basically. They would generally be making their ATP in glycolysis if they are fermenting a sugar, but they could also be making their ATP from coenzyme A derivatives. If they're fermenting a sugar, then they're going to be producing butyric acid or butanol or acetone. So if the pH is more neutral, they're producing butyric acid. If the pH goes down, they're producing butanol and acetone instead. The ones that ferment amino acids would produce ammonia, carbon dioxide, and coenzyme A derivatives. So they're basically breaking that amino acid down to get the amino group and the carboxyl group, which is converted to carbon dioxide. And then the rest of it, the side chain and that central carbon, that's going to be converted to the CoA derivative. And a lot of those CoA derivatives have a really bad smell. So the smell of like rotting flesh, that is actually those CoA derivatives that are being produced by Clostridial bacteria doing amino acid fermentation. Then you have the nucleotide fermenters using nucleotides as their substrate. They are producing, for the most part, the same products as the amino acids fermenters. So ammonia, carbon dioxide, and coenzyme A derivatives. In the diagram, you have an example of a typical amino acid fermentation pathway, which again, we're not going to worry about the details here, but we're starting with a couple of different amino acids, alanine and glycine. The alanine is being oxidized and converted ultimately to acetate. The glycine is being reduced and converted ultimately to acetate. So this organism would have to do both of these pathways at the same time. The electrons are being transferred from alanine to glycine, and you need to drop them off at glycine in order for you to get back your NAD+. You're going to be generating ATP on both sides of this pathway at the final step when you're producing acetate, and you are going through CoA. So here's CoA, it's coming in, being used to make acetyl-CoA, that's converted to acetyl P, and finally to acetate. So this would be your CoA derivative right here. Well, actually all of these are CoA derivatives. Then you're also producing your carbon dioxide and your ammonia. Then we have propionic acid fermentation. Propionic acid fermenters are mostly found in the genus Propionibacterium, which is also the genus that is responsible for causing acne. So this is a bacterium that you could find living on your skin. Actually, though, it's an anaerobe, so it has to live in your pores. It cannot live on the surface of your skin. And it's fermenting propionic acid, well, producing propionic acid in fermentation for its energy, in a process that is also producing carbon dioxide as another fermentation product. These bacteria that do propionic acid fermentation are also the bacteria that are used to make Swiss cheese. So the little holes that you have in Swiss cheese are actually made by the carbon dioxide that they're producing in fermentation in addition to the propionic acid. For a substrate, they like to use sugars, and that can also include lactate that would be produced by lactic acid fermenters. So lactate is actually a sugar as well. And this is an example of somebody's fermentation product being somebody else's substrate. And the lactic acid fermenters are producing lactate or lactic acid. They're basically the same thing as a fermentation product, which is a waste product. But then these propionibacteria bacteria are taking that up and using it as a substrate for their own fermentation, or using a different sugar like glucose. So when they do use glucose as their substrate, then they're making their ATP in glycolysis, and then they go into a modified Krebs cycle or citric acid cycle. If they're using lactate, then they skip the glycolysis and they go directly to making ATP in a modified Krebs cycle or citric acid cycle. So in the diagram, you can see basically how it goes when they're using lactate as their substrate. So they're skipping glycolysis, but they are converting that lactate to pyruvate. Part of that molecule, part of that pyruvate is going to make acetate and carbon dioxide ultimately, but part of it is going into the modified Krebs cycle, being added to or converted to oxaloacetate. So this whole thing here actually is your modified Krebs cycle. So you're making a little bit of ATP there, and you're also ultimately converting that lactate to propionate or propionic acid. So up to now, we've talked about fermentations that involve substrate level phosphorylation for making your ATP. Most fermentation does, but there are actually some exceptions. Some examples of fermentation reactions in which the energy-rich compound that you're using as a substrate actually doesn't have enough energy for substrate level phosphorylation. It doesn't have enough energy to transfer a phosphate group to ADP, creating an ATP. So these organisms that are doing fermentation without substrate level phosphorylation are somehow able to still make ATP, even though there's not enough energy there in that compound to even make it. And they would generally do that by pumping ions. So an example is a species Propionogenium modestum, which is fermenting succinate as its substrate to propionate as its fermentation product. But there's actually not enough energy in succinate to phosphorylate ADP to ATP. Instead, you have enough energy in the succinate to operate an ion pump and pump a single sodium atom across the membrane. So ultimately, by doing this, you're going to make a sodium motive force, and then you can use that to make ATP using a modified ATP synthase. That's actually what's shown in this diagram right here. So here you have your succinate being pumped into the cell. Actually, I shouldn't say pumped, being transported into the cell. This succinate is going to be fermented to propionate, which would then exit the cell, and you have just enough energy in this fermentation to operate this pump and pump a sodium ion out of the cell, which eventually, if you keep doing that, is going to generate a sodium motive force, which could be used by a sodium ATP synthase to create ATP. So as long as you're getting enough energy out of that substrate to pump a single ion across the membrane, you can eventually pump enough ions to make enough ATP to run the functions of the cell. You also have some species of bacteria in the genus Melanonimus that are fermenting in a similar way, fermenting malonate to acetate. So their substrate is malonate, their products are acetate and carbon dioxide. But malonate, again, does not have enough energy to add a phosphate group to ADP, creating ATP. Again, they're just using the energy of this fermentation to pump a single sodium atom across the membrane, eventually setting up a sodium motive force and using a modified ATP synthase to make their ATP. Then you also have the species Oxalobacter formigenes, which is fermenting oxalate to formate, and again CO2. So their substrate is oxalate, and then their fermentation product is formate and carbon dioxide. This is a species of bacteria that actually is found living in your gut, and is thought to be helpful for you because oxalate can be toxic, and it's removing it and converting it to formate. Oxalate can actually be a component of kidney stones, so it's good to get it out of there. This is a little bit different. This species is not actually using their fermentation to pump a sodium ion. Instead, the reaction that's involved in fermenting oxalate to formate will actually require protons. So you're actually going to add some protons to that oxalate to turn it into formate and carbon dioxide, which means you're reducing the number of protons in the cytosol of the cell, and you're not reducing the number that are outside the cell, so eventually you're going to make kind of a weak proton motive force by doing that as well. And then that would be used by ATP synthase to make ATP. So all of these species are kind of living on the edge. What they're doing for energy is not great. They're not getting a lot out of it. They're barely getting by, but they are able to get by using these tiny amounts of energy in these low, not very energy rich compounds that are just energy rich enough to at least pump a single ion or to consume, to drive a reaction that consumes a proton from the cytosol of the cell. So they're going to have to do a lot of these fermentation reactions to make enough ATP to actually run themselves. The last type of fermentation that we'll talk about is involving syntrophy. Syntrophy is when you have two different microbial species that are working together to catabolize something for energy. And you need both of them there, both doing their part to actually do the catabolism and get energy out of it. Usually, what they're catabolizing is the fermentation product from some third species. So those fermentation products are always going to have less energy in them than the substrates that they were produced from. So for a lot of bacteria or archaea that are using a fermentation product from somewhere else for energy, that's not the most efficient process, and sometimes they're not even able to make it work unless they have another microbial species helping them. The two species that are involved in a syntrophy relationship are not going to be able to live without each other. Each of them is completely dependent on the presence of the other for their metabolism, which means that if you try to culture one without the other, it's going to be impossible. So that makes these guys kind of hard to isolate and culture, because you have to figure out which partner they require and culture that guy as well at the same time in order to grow them. But syntrophetic organisms are really important for the ecology of environments that do not have a lot of oxygen in them, or you have a lot of fermentation going on, a lot of different fermentation products being produced. There's different types of syntrophy. One of the most common types is interspecies hydrogen transfer, in which you have one species that is producing molecular hydrogen and another species that is consuming it as a source of energy, maybe as an electron donor. So of course, the species that is consuming that molecular hydrogen is benefiting from this relationship. It needs that other species to keep providing it with the hydrogen that it needs for its metabolism. But the first species that is actually producing the hydrogen is benefiting as well. That's because the reactions that produce hydrogen, that produce molecular hydrogen and energy, are really unfavorable reactions, which means it's very difficult to make them go and produce that product of molecular hydrogen. So if you look at the standard delta G for those reactions, it's very unfavorable. It's basically a positive number, which means that it's not a spontaneous reaction and it's not going to just happen. And when it does happen, you're not going to get energy out of it. Actually, you're going to have to put energy in, which means that these species are not able to produce hydrogen as a part of their metabolism if they're doing a reaction under standard conditions with the standard delta G. But one part of the standard conditions is having every reactant and every product at 1 molar concentration. So what the consumer of molecular hydrogen is doing here is eating up that hydrogen just as it's produced, which means that the concentration of molecular hydrogen will never rise. It'll never reach 1 molar. It's going to be really low because as soon as it's made, it's used by this other partner species. When you have very little of that product, that is going to change the delta G and drive it more negative so that it actually does become a favorable spontaneous reaction that you can use to get energy out of. If the consumer stopped consuming that molecular hydrogen as soon as it's produced, then you're going to have concentration of hydrogen rising, the delta G is going to change, it's going to go more positive, and the reaction will at some point not even be possible. So in the diagram, you have kind of an example of the interspecies hydrogen transfer. You have two species here. One of them is doing ethanol fermentation. The other one is doing methanogenesis. The ethanol fermenter is fermenting that ethanol to acetate, which also produces molecular hydrogen. And as you can see, the standard delta G for that reaction is positive, which means it is not spontaneous and it cannot be used as a source for energy. Then your other partner in this relationship is going to be consuming molecular hydrogen and using it to produce methane as part of their metabolism, which is a favorable reaction. So when you actually put these two guys together and let them live together, you see that the overall delta G for what they're doing is actually negative. So you have this very negative delta G for the methanogen and a positive delta G for the ethanol fermenter. And when you combine them, you get overall a negative delta G. So the total reaction would be converting ethanol to acetate, also producing methane. So the molecular hydrogen is actually kind of canceling out from both sides of the reaction there, so you don't see it, but it actually would be there, transferred from the first species to the second species and consumed immediately by that species to create the methane. Finally, we will talk about hydrocarbon metabolism, which is the last type of metabolism that we will cover, although it's not the last that exists. So organisms that are doing hydrocarbon metabolism are using hydrocarbons for energy as electron donors. A hydrocarbon is a very simple lipid. It's actually the simplest type of lipid. It only contains carbon atoms and hydrogen atoms. So it'll be like a chain of carbon atoms studded with hydrogens. You have two types of hydrocarbons. They could be aliphatic hydrocarbons or aromatic hydrocarbons. The aliphatic ones have a chain of carbon atoms studded all over with hydrogens, and then the aromatic hydrocarbons have that chain wrapped around and attached to itself to form a ring. So here you have an example. So here's some aliphatic hydrocarbons, just really simple aliphatic hydrocarbons. They can actually get really long, so these chains could be super long. And here's a simple aromatic hydrocarbon that's forming a ring. And you see that in all of those, the only atoms that you have are carbons or hydrogens. So whenever you're using hydrocarbons for energy, in general, no matter if it's an aliphatic or an aromatic hydrocarbon, you have to oxygenate it first by adding oxygen atoms to that hydrocarbon. And there's two ways to do that. It could either be aerobic or anaerobic. If you're adding oxygen aerobically, using actual molecular oxygen from the air, you're going to also use oxygenase enzymes. You have two basic types of oxygenase enzymes. Monooxygenase is able to add a single oxygen, so it'll actually split molecular oxygen in two and add a single oxygen to your hydrocarbon, and it also requires an electron carrier in order to function. Dioxygenase enzymes are going to add both of the oxygen atoms in molecular oxygen, and they do not need an electron carrier. On the other hand, if you're oxygenating your hydrocarbon in an anaerobic fashion, you're not going to use molecular oxygen or oxygenase enzymes. Instead, you're going to take some other compound that already contains oxygen and just transfer those oxygens over. A very common example would be fumarate, which is shown right here. So it's like a skeleton of carbons with a couple of hydrogens and then a bunch of oxygens stuck to its ends. After you have oxygenated your hydrocarbon, you're going to end up with something that can feed into a respiratory path, usually into the Krebs cycle or citric acid cycle, sometimes into pyruvate oxidation instead. So if you're doing an aliphatic hydrocarbon oxidation for energy in an aerobic fashion, you are first going to be using a monoxygenase enzyme to add oxygen to those hydrocarbons, which then produces fatty acids. A fatty acid is actually just a hydrocarbon that has a carboxyl group on it, so you really just have to add a couple of oxygens to that hydrocarbon in order to make it a fatty acid. That would be done by monoxygenase. And then after that's oxygenated, you're going to use a process called beta oxidation to break it down and catabolize it. Beta oxidation for eukaryotes is actually going to happen inside the mitochondria. Of course, for prokaryotes, just in the cytoplasm. And it involves breaking that fatty acid up piece by piece into chunks that are each 2 carbons large. Each of those 2 carbon chunks is then going to go to create an acetyl-CoA. So those 2 carbons are going to be added to coenzyme A, and that gives you acetyl-CoA since acetate has 2 carbons. And you also are going to have electrons from those chunks that are going to NADH. So then that acetyl-CoA is just going to enter the Krebs cycle, and the NADH is going to enter the electron transport chain, and you're going to make ATP in the same pathways as you would for glucose and respiration, basically, except that you're feeding into the Krebs cycle and electron transport chain in a different fashion. If you are doing this anaerobically, then the basic mechanism is the same. The main difference is that you are not oxygenating it using the monoxygenase. Instead, you are transferring oxygens from something like fumarate. This diagram here is summarizing that. Again, you do not need to know this diagram, and we're not going to go over the details of it too much. But on this kind of side of the diagram, you're starting with a hydrocarbon and you are oxygenating it aerobically using molecular oxygen here and a monoxygenase to add the oxygen. Ultimately, that is going to give you this product here, in which you have a carboxyl group added to that hydrocarbon, which means that it's now a fatty acid. Then you can break it into your 2 carbon chunks. So the main bulk of the molecule would be over here. It can be quite long, actually. This one that we started with actually had 8 carbons total. So now this would be, about 4 more carbons over here. This end of the molecule, where you have the carboxyl group, is actually going to end up being attached to CoA, and at that point it's basically an acetyl-CoA that is just waiting to be chopped off. So you chop it off, there's your acetyl-CoA. This is going to feed into the Krebs cycle. Then you have the aromatic hydrocarbons, which are going to be oxidized for energy in a completely different fashion because they are rings, not chains. So you can't use beta oxidation to break them up into little 2 carbon chunks, and each chunk turns into an acetyl-CoA. If you're doing this aerobically, to oxygenate them aerobically, you're going to have dioxygenase enzymes that convert the aromatic hydrocarbon to a catechol. It actually could be converted to different things, but usually it's a catechol, or usually it is catechol. Catechol is right here. So here's your hydrocarbon benzene that you're starting out with in this example, and you're going to have oxygenase enzymes. In this case, actually it's a monoxygenase, but it would generally be a dioxygenase that is adding oxygen to it. Ultimately, you need to add 2 oxygens, and that's going to convert it to a catechol. Once you've made your catechol, or a compound very similar to catechol, you can catabolize that. You're going to have different enzymes that are able to split that up into different types of compounds, so depending on which exact aromatic hydrocarbon you started out with. So some of them are going to end up looking like pyruvate, some of them are going to end up looking like acetyl-CoA, or some intermediate of the Krebs cycle. So if you end up producing or changing catechol into a compound that looks like pyruvate, then it's going to enter pyruvate oxidation. If you convert it into something that looks like acetyl-CoA, or a Krebs cycle intermediate, then it's going to enter the Krebs cycle and proceed from there along the normal path for respiration to make ATP. you're actually going to be using oxygenase enzymes to do that conversion as well. If you're doing this anaerobically without oxygen, then your goal is still the same. You're still going to be converting this aromatic hydrocarbon into something that looks like pyruvate or acetyl-CoA or a Krebs cycle intermediate. It's going to be feeding into probably the Krebs cycle, but the exact way that you do it is going to be different.